Welcome to Science Research Weekly, Episode 8, Flash Grants in the Pan. Get ready for technological tribology, tingible translational cell biology, flipping fossils, volume 14.4 of the R Journal, ebook tips and tricks, and flash grants to fixate on. This episode covers March 3rd to March 9th. Let's get cracking with some research articles. I scoured the news from Science Daily and I have 5 articles lined up. First, we have novel friction stabilization technology for surface damage conditions using machine learning. Here, researchers worked on friction control technology that combines morphable surfaces and AI to reduce friction through active adaptation. Fun fact, the study of friction is called tribology. Something I learned a month ago listening to a 3-minute thesis competition. The more you know. Moving along, we have the paper crevasse refreezing and signature of retreat observed at Kamb ice stream grounding zone. So using a diving robot called the Ice Fin, researchers examined crevasse melting and refreezing under the Ross ice shelf, offering an unprecedented look. Moving right along, we have apoptotic cell fragments locally activate tingible body macrophages in the germinal center. So, tingible body macrophages, these are from lymph nodes. These are rare cell types that clean up the immune system's own waste, namely motile apoptotic cell fragments. Therefore, they're an important but enigmatic portion of the cellular maintenance system. It took over 140 years to have the technology to look at them in detail. Learning more about such cells could help understand the causes of some autoimmune diseases and provide clues in helping treat them. Another fun fact, tingible means capable of being stained, so for imaging. We're learning all sorts of words today, aren't we? Fourth on our list is dynamic interplay between RPL3 and RPL3 containing ribosomes modulates mitochondrial activity in the mammalian heart. So researchers found that the heart and skeletal cells connecting to the heart have a different ribosomal protein than other cells. So they have RPL3L rather than RPL3. Fascinatingly, in high demand situations, heart cells replace those RPL3L3 ribosomes with the typical ribosomes. Last on our overview of research articles, I have an abundant sea anemone from the Carboniferous Mason Creek, Lagerstadt, USA. So it turns out a very common fossil, Seszelia, maybe not commonly pronounced. Anyways, it has a nickname called blob. These fossils found a lot of places, especially Lagerstadt. It's not a jellyfish, as was long thought. Instead, by flipping it around, researchers realized it was a sea anemone, a soft body organism that until now was very hard to find fossilized. In the Science Daily article that covered this paper, one of the best quotes I've ever heard came from James Hagedorn, the study's co-author, in reference to the fossils found in the Lagerstadt site, quote, "These fossils are better preserved than Twinkies after an apocalypse." There you have it. Anyways, there were some articles that didn't quite meet the grade to go through in any detail, but I'll note their names in case you want to delve into them yourselves. There's Plant Roots Fuel Tropical Soil Animal Communities, computationally guided DIW technology to enable robust printing of inks with evolving rheological properties, absorptive exchange of coccolith biominerals facilitates viral infection, and memory of stochastic single-cell apoptotic signaling promotes chemoresistance in neuroblastoma. With that being done, let's move over to research tools. So, Python 3.12.0 Alpha 6 just released, the 6th of 7 planned alpha releases for 3.12. Under the "and now for something completely different" section, get ready for Shakespeare. Volume 14.4 of the R Journal is now available online. Here are some of the articles I found most intriguing. There is NetGWAS, an R package for network-based whole genome association studies. There is GGLM-EXT POISE, advances in dealing with over and under dispersion in a double GLM framework. Making province work for you. Tree search, a morphological phylogenetic analysis in R. Bayes PPD, an R package for Bayesian sample size determination using power and normalized power prior to generalized linear models. 5 new ARC packages from CRAN to get you cooking with gas. There is BayesFluxR, implementation of Bayesian neural networks. Lorem, generate Lorem Ipsum text. HDRFA, high dimensional robust factor analysis. Guildai, track machine learning experiments. And aihuman, experimental evaluation of algorithm-assisted human decision making. Got our bloggers on the brain? Well, I sure do. Here are 2 posts to preen about. First, there are 7 new books added to the Big Book of R. 1, Population Health Data Science with R. 2, Reproducible Analytical Pipelines, Masters of Data Science. 3, Applied Statistics with R. 4, Comparative Methods. 5, a course in exploratory data analysis. 6, Probability and Bayesian modeling. And 7, R without statistics. Interested in making an ebook? Well, Bruno Rodriguez from Econometrics and Free Software has some tips and tricks on how to generate an EPUB that passes EPUB check using Quarto. With our tools set aside, let us contemplate research funding to finish out. A variety of grants caught my eye. Eye of the beholder, perhaps, but funding all the same. The Department of Defense has a Research Collaboration Award, aiming to create basic research collaboration between a new PI and a co-PI who's had a DoD award. This is for the Defense Established Program to Simulate Competitive Research, or DEPSOR. The DoD also has 2 awards for ovarian cancer, 2 more awards. There's Clinical Translational Research Award and then a Clinical Trial Award. There were 2 notices of special interest from the NHBLI, the National Heart, Lung, and Blood Institute, one for research project grants in valvular heart disease, and another in integrative omics analysis of the NHLBI top med data. The Department of Health and Human Services has small R01s for clinical trials targeting diseases within the mission of NIDDK, that is the National Institute of Diabetes and Digestive and Kidney Diseases. Now then, Biogen has a sponsorship funding for a variety of diseases and focus areas. Aiming big? Well, the American Heart Association, AHR, is funding strategically focused research networks of 3 to 5 centers over 4 years for a total of $12 to $20 million. So grab every single one of your friends to apply for this. Your friends who happen to be research institution heads. Now then, the Cancer Research Institute, CRI, has a Technology Impact Award to address the gap between technological development and clinical applications of cancer immunotherapies. Then, the Institute of Advancement for Food and Nutrition Sciences, IAFNS, has a gut microbiome committee which released a grant opportunity on the investigation of the relationship between live dietary microbe intake and health outcomes. And lastly, but certainly not leastly, North Carolina Life Sciences has flash grants available for cycle 3 in all areas of life sciences except Class 1 medical devices and consumer products. So flash grants support short, tightly focused research projects in these 2 categories. 1, to obtain initial exploratory proof of concept or feasibility data necessary to suggest the disruptive potential of a life sciences technology. Or 2, to conduct the final experiment or experiments needed to advance a basic life science research program into the translational research phase of development. So awards are up to $20K or $27K depending on the R01 status of the university applying, or PI from the university applying. They are due March 22nd, so make like the Flash and get going. Well, out of the frying pan into the fire. Thanks for joining me for Science Research Weekly. Science on.